Hello, everyone. So, welcome to this new global IMMUNOTALK, and it's a great pleasure for me, Anna Maria Lennon, to introduce our speaker of the day. So, this is Ziv Shulman, and he has been an associate professor at the Weizmann Institute of Science in Israel since 2015. So, a few words about Ziv's career. He had already started his research career actually at the Weizmann Institute, where he did his PhD in the lab of Ronan Allen. And this is probably when he developed strong affinities with live cell imaging while he was studying the crawling of T lymphocytes and their interaction with endothelial cells under mechanical constraints. So in this specific case, shear stress. So his PhD work was very productive, actually, and it allowed him to show, between other findings, the role of intracellular vesicles from endothelial cells in this process, as well as the important player integrin LFA1. And Ziv also described the involvement during his PhD of the RAC-GTP exchanging factor DOC2 in the migration of T lymphocyte, which had not been discovered yet. So from his PhD, he moved to postdoctoral training to the US, New York, Rockefeller University in the lab of Michel Nussenswag. So there, he devoted his work to the study of antibody affinity maturation in germinal centers. And he used for this, again, microscopy approaches. And his main contribution was to describe the key role played by signaling from follicular T cells in this process. So in 2015, as I initially said, Ziv joined the Weizmann Institute as a faculty, and he followed up on his postdoctoral work, investigating the molecular and cellular pathway that drive the production of protective antibodies and that allow the establishment of long-lasting humoral immune responses. So Ziv Lab is particularly interested in understanding how germinal centers function within complex microenvironments. So for example, has made a beautiful contribution showing that how BCR affinity controls the colonization by B cells of the domes in pair patches and the entrance of these cells into germinal center. And lately, he has also been described B-cell dynamics in the context of pathologies, including infection and cancer. And here I will mention a study where Ziv's lab discovered that tumor-reactive autoantibody very often derive from autoreactive B-cells precursors. So given all his contributions, he has obtained several honors, including the HFSP Career Award grant to create his lab. He has obtained 2 ERC grants for young investigators, and he's a member of the EMBO Young Investigator Network. Importantly, recently, he was awarded the Rappaport Prize for Promising Scientist. So we look forward for Ziv talk, but before we start, I must ask him one of our favorite questions. So Ziv, what has been your most surprising scientific finding and how did it impact your research path?
Hi. So thank you for the kind of introduction. So about the question, so I had a study like that I'm going to speak about in a couple of minutes, where we study B-cell immune response in the nose, and I had a very talented student that compared lymph nodes in the nose to lymph node in the gut to pair patches, and like she worked on that for several years, and that was the most boring project in the lab. She couldn't find anything special. Like the lymph node of the nose looked exactly like the lymph node in the gut. And I gave up on that. So I told her, okay, let's write a paper. Let's just publish whatever we have and move on. And also I felt bad because she was a very talented student and I wanted to do something with more impact. But nonetheless, she wanted to continue working on the problem. And I started to write a paper. We disagree. And then like one day she shows up in my office and she showed me a picture and she shows me B cells inside of the bones of the nose. And I look at that and I say, oh man, like that's amazing. Like that looks like a nature finding. And so that was a discovery which really surprised me, and I wanted to give up, but she didn't. So what I learned from that, or what to be learned from that, is that persistent is important, but even more important is to get students which are more persistent than you. So this is the story that I have to tell about that.
So thank you, Ziv, for sharing this story and telling our students to be disobedient. It's very important.
Yeah.
And now I will invite you to share your screen. Please go ahead, show us your science.
Yeah. Okay. Okay, so thank you very much for having me today. Thank you for the kind introduction by Anna Maria. Um, so I put here a picture that we took in the lab. We took a photo of a lymph node. You can see the original picture. This is a human lymph node, and I uploaded this picture to ChatGPT, and I asked ChatGPT to make the picture look like Starry Night, Van Gogh style Starry Night picture. And this is what we got. And here you see the structure of the lymph node, and you see these circles over here, these yellow circles. These are germinal centers. You can see them here in how it looks like in real and how it's nicely look in this picture. And what I want to say is that my lab studies germinal centers. Everything that we do is related to germinal centers this way or another, either events that occur before germinal center response or after the germinal center response. But we also always go back and ask how the germinal center contribute to immune responses. So we study germinal centers we ask basic questions, how the germinal center work. But what I also like to do, I like to complicate things by asking how germinal center function in complex microenvironment. And when I say complex microenvironments, I mean mucosal tissues where you have bacteria and other factors that play a role, but also in cancer where B cells are entering tumors, but what they are doing and what are the function and what the importance and how it's relevant to the disease is more complicated. And what I'm trying to do is to take the rules, the basic rules that we learned from mice immunized with NP-OVA and simple models, and see if they are apply to more complex situation, and also discover new rules or new biology in this complex situation. Okay, and yeah, and we also sell, we have merchandise with this picture. Okay, so we study germinal centers. Here you see a picture of my kid getting a corona vaccination. When we get vaccinated or when we get exposed to a pathogen, pieces of the pathogen or pieces of the vaccine are being drained into lymph nodes. Now, within lymph nodes, there are many cells, mostly T cells and B cells, and some rare cells will express a receptor that can bind the antigen that was derived from the pathogen or from the vaccine. So some rare B cells express an antibody on the surface that can interact with pathogen-derived or vaccine-derived antigens. And then these B cells start to proliferate. They can differentiate to different cell types, and they can seed germinal centers. And these are the germinal centers that you see over here. This is a Ki67 marker, and these are proliferating B cells within the lymph nodes. What is the point of the germinal center? What is the purpose of the structure or the germinal center reaction? So the purpose is to undergo clonal expansion. You make more B cells with antigen-specific receptors. The B cells increase their antibody affinity, And also the output of the germinal centers are plasma cells and memory cells. These cells provide us with long-lasting immunity. So now if we meet the antigen, the pathogen again, we will be protected by antibodies that were made by these plasma cells, which originated from germinal centers that were formed previously. Um, so, um, so now I'll explain in a simple way how the germinal center function. So B cell encounter an antigen, and then B cells go into the germinal center response. And within the germinal centers, B cells can mutate their antibody encoding genes. So mutations are introduced into the B cell receptor, the immunoglobulins, and we have now a different sequence with mutations, and this sequence can be something that enhance antibody binding of an antigen, or it can make it worse. So then there is a selection phase in which the best B cells, or B cells that bind antigen better than the others, they are being selected, and these B cells can go back and mutate again. And this is kind of a cyclic process in which there's a mutation, and then there is selection, and then mutation again, and selection again. And this process basically push the affinity of the antibody higher and higher over time. So this is kind of evolution. This is a mini evolution that takes place in germinal centers within lymph nodes. During this process, memory cells come up, also plasma cells. Plasma cells can leave the germinal center, or pre-plasma cells leave the germinal centers and become plasma cells. These are long-lived cells. They secrete antibodies. They constantly secrete antibodies without the presence of the pathogen, and basically they give us immunity. So once a pathogen wants to come in again, it will meet these antibodies, and they're supposed to prevent the entrance of the pathogen into our body. So this is a very simple scheme. Obviously, things are a little bit complicated. Germinal centers are guided by T cells. So in order for B cells to make germinal center, it has to interact with a T cell, and the T cell provide the B cell with help that promote germinal center seeding. Then the B cells seed the germinal center, and then it can proliferate in an area that we call the dark zone. And the selection of high affinity variants is mediated by T cells in the light zone of the germinal centers. The selection is made by antigen presentation. So B cells with a good antibody and effective antibody can uptake antigens and present it to T cells and get a strong help signal from the T cells, and then they can proliferate. B cells which are less effective will pick up less antigen, present less antigen, and proliferate less or not proliferate at all. So the whole germinal center response is guided and controlled by T follicular helper cells, T cells that guide the B cells and tell them what to do. Okay, so these are pictures. In my lab, we know how to image the germinal center response and image T cell B cell interactions. I will show you a nice example. So what we do, we take mice, we put fluorescent cells into the mice, and then we expose the lymph node, and we put the mouse under the microscope. This is a 2-photon scanning laser microscope that can penetrate deep into the tissue, and we can visualize the immune response. And here we look into the mice. You see here cells moving around within the lymph node during germinal center seeding, and they can interact with each other. We can zoom in on interaction. So here you see a T cell in red and a B cell in blue. So you see that they are coming together. So we can visualize that. We can quantify the interaction. And we can also ask questions. So here specifically in this study, we knocked out ICAMs from B cells. And when we looked at T cell B cell interactions, when ICAM is removed, we could show that the interactions become shorter. So here you see a long-lasting interaction, about 50 minutes. This interaction is shorter. It's about 15-minute interaction. And there are implications about germinal center seeding when this interaction is shorter. So this is the way we use 2-photon microscope. There are limitations to this approach. We see a small field of view. We can scan the whole lymph node, but nonetheless, the penetration is limited. So we looked for a way to visualize the whole immune response, to see all of the germinal centers and all of the B cells within a mouse. So for that, Liat Stoller-Barak, she's the staff scientist in my lab. She used light sheet fluorescence microscopy and tissue clearing, and basically she takes a lymph node of an immunized mice with reporter for B cell activation, and this allows us to visualize all the responding B cells within a lymph node. And here in green, you see single cells. These are B cells that came out from the germinal center or just become activated. And you see these clumps of cells. These are the germinal centers. These are many, many cells that are found very close to one another, so we don't see single cells. And this approach allows us to see the whole germinal center structure. So we see this area over here, and you see that it's quite smooth, and it faces the inner side of the lymph node. And you see this area over here, which is a kind of a rough area. You see budding cells from this side. And by looking at this structure, we see that the germinal center is polarized, and this is supposed to be the light zone because it faces the lymph node capsule. And basically, this roughness suggests that cells are coming out from the light zone, that the light zone is the primary site of exit in germinal centers. Nonetheless, this is fixed sample, so we don't see cell leaving. We just got an indication that this is what happens. And in order to really nail down this conclusion, we use photoactivation. So what we do, we label cells within the light zone. So here you see this is marker for follicular dendritic cells, a marker for the light zone. We label the cells, we turn on the photoactivation, and then we track them over time, and we can find cells that move out from the light zone into the lymph node cortex. And it's something that we can quantify over time, and we can see that cells are moving out. In the dark zone, when we label the dark zone, we do see cells coming out, but they are very rare. Most of the cells return to the light zone after 4 hours, and we saw very few cells going out. You can see the quantification over here. So light zone is the primary exit site from the germinal centers. I'm not saying there is no exit from the dark zone. It can happen, but using this approach, we could show that the major exit site is the light zone in the germinal center structure. Okay, so back to the whole organ imaging and tissue clarity, we realized that we can use this approach to visualize multiple niches within lymph nodes, and we use the light sheet in order to image pair patches. Pair patches are the lymph node of the gut, and there are two B cell niches in the pair patches. There are the germinal centers and the B cell follicles, but there is the subepithelial dome, where antigen is being delivered to B cells, and this is where B cells become activated and class switch to IgA. And if we use typical fluorescence approaches, we can see the germinal centers, but it's rarely that we can catch both the germinal center and the subepithelial dome in the same shot. So here this project was led by Adi Birama, a former PhD in the lab, and she used the whole ear organ imaging approach to image pair patches, intact pair patches. And what was nice about this approach is that we could image all the B cell niches. And here you see the subepithelial dome, this area over here. So we see B cells in this part, and here you see germinal centers, and the cells are moving from the subepithelial dome into a germinal center. We made a time course, and I'm not showing that, but this is the direction of the movement. So we see both the compartments, and thus allowed Adi to ask questions about what regulates the movement between these two compartments, and the nicest result was about affinity. So if we take high affinity B cells and image them, they go into the subepithelial dome, and then they immediately seed germinal centers. And this movie is very similar to the previous movie that I showed you. However, what was very interesting is that if we take low affinity B cells, express low affinity BCR, they can respond to an antigen in pair patches and expand, but they get stuck in the subepithelial dome. So here you see a subepithelial dome, but there is no seeding of germinal centers. So this approach allows us to get to the conclusion that there is affinity-based checkpoint in the subepithelial dome, and only high affinity B cells can continue and seed germinal centers, whereas the low affinity counterparts, they get stuck in this area. They do respond to an antigen, but they just don't go on and seed germinal centers. Okay, so we had this technology, and that was pre-COVID times. And then COVID started, and people become very interested in immune response against airborne pathogens, and also the need for development of nasal vaccine came up. And we realized that we have the tools to image the immune response, and we realized that that's useful and important because back at the COVID time, there were many studies published about antibody-mediated immune response in the nose against COVID and against also against all kinds of vaccines. But the studies always quantify antibodies and different types of antibodies, IgA, IgG1, and also affinity maturation, but it was all about antibodies. And we were wondering, where are the cells that make the antibodies? Where are the germinal centers that make the antibodies? Our germinal centers are important to make these antibodies. So we wanted to visualize the immune response against nasal vaccination within the nose. And we realized that we have the tools to do that. We did it in the gut, so there was no reason that we won't be able to do that in the nasal compartment and the upper airways. So I was speaking about pair patches, and in the upper airways, we have a different type of lymphatic structures. In the mouse, the lymph node of the nose, or the lymph node that is closest to the nasal cavity, is known as nasal-associated lymphoid tissue, or NALT in short. So if you look at the nasal cavity here, you see the NALT, the nasal lymphoid organs. If we zoom a little bit in, these nasal tissues contain germinal centers, and there are subepithelial dome here, so they look very much like pair patches. And in fact, it's like a small pair patch that is found in the nose of the mouse. And to the best of my knowledge, it's the only other organ other than pair patch that have subepithelial dome can support class switch to IgA within the mice. So we focus on this lymph node, and we study immune response germinal centers within these lymph nodes. So this project was led by Jingjing Liu. She's a PhD student in the lab, and she designed a vaccination approach that allows us to visualize antigen-specific B cells within nasal lymphoid tissues within mice. And basically, she transferred B cells, antigen-specific B cells, B1 high, they are specific for neutrophenil, for NP. We also can transfer T cells, so we can also look at the antigen-specific OT2 T cell response. So she takes these cells, she transfers them into mice, and then she immunized the mice intranasally with NP-OVA. And after a few days, we used the two-photon microscope in order to visualize B cell immune response within the NALT. Now, there's important factor here. We use MPLA as an adjuvant. MPLA is a synthetic LPS, and it's important to note like LPS is usually used as an adjuvant for nasal vaccination, and that's not very useful. I'll show you why. So in 2022, we had a study where we looked at immune response against bacteria and also against LPS, and we found out that if we use LPS as an adjuvant, it doesn't give a very strong germinal center response. So if we use alum, we get a nice germinal center response. If we use NPLA, we get a nice germinal center response. But if we use LPS, we don't. And that's even kind of surprising, because both MPLA and LPS are TLR4 ligands, but LPS is also toxic in some way, and MPLA is not. In fact, in this study, we figure out the mechanism. We found that monocytes goes into the lymph node, and this monocyte disrupt germinal centers. when we use LPS. When we use MPLA, this doesn't happen. So this is the reason we use MPLA for vaccination, and I recommend if you do nasal vaccination, MPLA is way better than LPS. Okay, so Jingjing immunized the mice, transferred the cells, immunized the mice, and then we saw that B cells can colonize the subepithelial dome, and we get strong expansion of B cells in the subepithelial dome within the nasal lymph node. Here you see pre-existing germinal centers. We use AID GFP mice, so these germinal centers are specific for bacteria, and these germinal centers are specific for the vaccine. And then after 10 days, after vaccination, the subepithelial dome response is gone. The B cells invade pre-existing germinal centers, and also you see new germinal centers within the NALT of the mice. So we could visualize the B cell immune response within the NALT. So we did a lot of experiments here and time course analysis and visualize the T cells, and we have lots of data about the NALT, but there was one mystery that we couldn't solve for a very long time. And so we see this strong immune response in the nose, but to where the cells are going. So plasma cells support to be formed here, and memory cells, but for a very long time we couldn't find the destination of these cells. Where are they going? So Jingjing looked at many organs in the mouse. She looked at the bone marrow. She found very few B cells within the bone marrow. She also looked at the lung. I thought the cells are going to colonize the lung, but we didn't find any plasma cell in the lung from this vaccination. Also, the gut mucosa and vaginal mucosa, we didn't find any cells that seed the mucosal tissues. So we didn't find the cells, and that was very frustrating for a very long time. And then came out this paper. This is a really beautiful paper from Ashley Mosman lab, where he described that circulating antibodies in the circulation in the blood, they cannot reach the olfactory epithelium that is close to the brain within mice. And he described the mechanism in which plasma cells have to migrate into the olfactory epithelium and secrete antibodies locally and protect against invasion into the nasal epithelium by making a local antibody immune response. And that's a very nice concept, because when you think about antibodies or you think about plasma cells, usually they reside in the bone marrow or kind of organs, they secrete antibodies. And you don't need plasma cells in site of inflammation or in site of virus invasion. But here he demonstrated that in this context, it's important to get the plasma cells into this barrier and make a local immune response that protect the olfactory epithelium. So we got really excited about that, and we thought perhaps we need to look for plasma cells in this part of the mouse, in the olfactory epithelium, and also in the upper airways. So Jingjing went to the neuroscientist, and she learned how to cut the mouse head. And here you see a section of the mouse head. Here is the brain, and here you see the nasal cavity. Here you see the NALT over here. So this is the nasal tissue that I showed you earlier, the lymphatic tissue. And here you see these bones, which are known as turbinates. This is the bones of the nose, inside the nose. If we zoom into these bones, they look very much like a gut mucosa. So here we have a epithelium over here, and there is a mucus layer, and then there is a lamina propria that contain immune cells, and also there is a bone or a cartilage over here, and here there is something that looks like a bone marrow. I'm not sure it's a classical bone marrow, but nonetheless it really looks like a bone marrow. So it looks like a gut mucosa, but on top of a bone within the mouse nose. And this gut mucosa and epithelial cells, it contains many features that are associated with mucosal surfaces. There are tuft cells over here, and also goblet cells over here that secrete mucus, and also ionocytes can be found in this epithelial cell. So this is a classical mucosal epithelial cells of the upper airways. And then, so Jingjing took mice, she made her protocol, she transferred the cells, she immunized the mice, and then she cut the head of the mice, make section, and look in this area of the mouse. And surprisingly, she found antigen-specific cells, B18 high, that we transferred to the mice. We found it in the turbinates and also next to the turbinates in mucosal glands and also under the epithelial cells. She could find antigen-specific B cells within the mouse nose, and that was very exciting. Finally, we found the B cells that we transferred. We found to where they are going, and I'm not showing you all the data, but these are IgA secreting plasma cells within the mouse turbinates and in the area, in the gland area next to the turbinates that we could find. So now we have B cells in two locations. We have in the NALT and we have B cells in the turbinates. And now we wanted to connect these two finding. Wanted to know if these germinal centers and this subepithelial dome reaction make B cells that go into the turbinates. So to address this question, what Jingjing did, she ablated the NALT by using a hot wire. So she put a hot wire into the mouth of the mice and ablated the turbinate, or she did a sham experiment where she put the wire next to the turbinate. And then she immunized the mice and took out the cells from the turbinates, and she could find B cells within the turbinate. These are B18 high cells that were transferred into the mice, and we could find B cells in the turbinates by fax. When we ablate the NALT, we don't find any B cells in the turbinate. So this is a proof that the turbinates is connected. The turbinates is connected to the NALT, and there is connection between these two organs. And then we wanted to know how this connection works. So we speculated that the cells are going through the blood. This is the way that immune response work in the gut. So there was no reason not to think that it works also in the upper airways. So what Jingjing did, she blocked migration through the blood. We use anti-PSGL1 antibody. So PSGL1 is a receptor that mediates lymphocyte rolling in sites of inflammation. The lymphocytes roll on the endothelial cells and then they cross the endothelial cells. So she used an antibody that blocked lymphocyte rolling, anti-PSGL1. And when she did that, the migration into the turbinate was blocked, which basically proves that the cells are going through the blood into the turbinates area. So the bottom line from this slide is that the NALT provides cells provide cells that home into the turbinates area for the blood. And then I wanted to know what chemokine signaling are directing the cells into the turbinates. So we did all kinds of different types of immunizations. And then I told JJ just take the turbinates as they are, no cell isolation, just take the tissue, smash it, make RNA, and let's do bulk RNA sequencing and try to find clues which chemokines direct the cells into the turbinates. And this is what she did. And we sequence B cells, we sequence the tissue, RNA from the tissue. We found inflammatory molecules such as ICAM1. It goes up in the turbinates in response to the immunization. Also NF-kappa B goes up. We found PIGR. This is a molecule that transports IgA antibodies into the lumen. So we were also happy to find this molecule. And then when she did co-terms analysis, we could see that adaptive immune response goes up in response to the vaccination. Also, B-cell immune response was up. So that means that we are in the right direction and we got a really adaptive B-cell immune response within the turbinates. And then we looked at the different chemokines that came up. So several B-cell chemokines came up, like CCL20 and also CCL28. So CCL28 was previously described as a mucosal chemokine in the gut, and now it came up in the turbinates. So we suspected that this chemokine might play a role in directing B cells from the NALT or direct B cells to go into the turbinates. We also find CCR10. So CCR10 is the receptor of CCL28, so that also makes a lot of sense. So both of them went up in the vaccinated mice. So we wanted to confirm that by a flow cytometer. We made a FACS analysis for CCR10, and all the cells within the turbinates express CCR10, and so that also makes a lot of sense. We also looked at CCR9. It's another chemokine receptor associated with the gut, and it's barely expressed in the cells that are found in the turbinates. Okay, so we have a chemokine and we have a chemokine receptor, and now do they play a role? So to address this question, what Jing did is to inject an antibody that blocks CCL28. So in control conditions, we can image immune cells in the NALT. These are B1-8i, antigen-specific cells in the NALT, and then we can find them in the turbinates as well. When JJ inject anti-CCL28, we found reduction in amount of cells that go into the NALT. This is with imaging. It's quite difficult to quantify that with imaging, so we did flow cytometry. And in flow cytometry, we can find the cells. And then when we add anti-CCL28, you'll see that they are gone. And this is quantification. So we could show that CCL28 participate in directing B cells from the blood into the turbinate. So that's the chemokine. So to summarize this study, what we discovered here is a new connection between a lymph node and a target site, the turbinate. The cells are going from the NALT into the blood and enter the turbinates. I showed you that B cells undergo expansion in the subepithelial dome of the NALT, and didn't show you that they express IgA, but they do. And then the B cells home to the turbinates for the blood, and CCL28 is the chemokine that facilitates recruitment of NALT cells into the turbinates. Okay, so this is a study about B cell in mucosal immune response, which I consider more complicated than studying B cells in peripheral lymph nodes. And now I'm going to speak about another complication. So now I'm switching gears, and I'm going to speak about B cells in cancer, and I'm switching from mouse to human disease. So my lab has been studying B cell in cancer for quite a long time. We started in 2016, like we had a project ongoing with lots of ups and downs, and it was a very difficult project. Also, we didn't get funding for this project for a very long time. And then like in 2020, there were 3 papers in Nature describing B cells in cancer. And basically, the take-home message of all of these papers was that the presence of B cells in tumor is correlated with longer patient survival, and also response to immunotherapy is better if tumor hosts B cells or host tertiary lymphoid structures. And we were very excited that these B cells, these studies were published in Nature. There are many studies that came after that, and also there was eventually in 2023 there was a specific meeting about B cell in cancer, in which like the importance and the role of B cells in cancer were highly emphasized, and showed that B cells do play a role in cancer, something that was neglected for many years. So my lab studied B cell in cancer during this time, and we focused on very specific question. We wanted to know if the B cells in the tumors are carrying an antibody which is specific for the tumor. And in many studies, people look at B cell, look at tertiary structure, but it's very difficult and it's very challenging to study B cell specificity, and that was our challenge. This is what we tried to prove. We wanted to know if the B cells are specific for the tumor. That's one question. And then obviously everything in my lab goes back to the germinal centers. And we wanted to know if germinal center contribute to anti-tumor immunity. So these are the questions that we focused in our study. And I will briefly show the study that we published about B cell and cancer, and then I will move to new unpublished results. So we chose to study ovarian carcinoma. The reason for that it was a Brad Nelson study that was published a while ago. This was one of the few studies that we had that gave us a direction. It described that CD plus, if you find CD20 plus B cells in ovarian carcinoma patient, the likeliness that they will survive longer is higher. So this is the study that we had when we started the project. So we decided to study ovarian cancer. And the student that led this study is a physician, and he was a PhD student in the lab. And it was also a collaborative studies with Irit Sagi, and the student was a shared student between us. So we went to physician, to the clinic, and I found partners, Ram Eitan and Oded Rabban from Rabin Medical Center, and they collaborated with us, and they provide us with tissues from ovarian cancer patients. These patients don't survive very long. It's a terrible disease, and the expectancy of survival over 5 years is very low. It's about 20%. So they provide us tissues from these patients that undergo surgery. We got pieces of these nasty tumors into our lab. We also get lymph nodes. We also get ascites fluid. So we got lots of biological materials that allows us to study B cell response in this cancer. The first observation that we had is that we took these tumors and we just stained for antibodies, and we found in many patients that the tumor are coated with antibodies. So this is a simple analysis, just stained with anti-IgG, and we found that the tumor cells are being coated, are attacked by antibodies within the patients. And so when we saw that, we did a screen for many cancer, any cancer that we could receive samples from the clinic. We tested for tumor coating, and we found that the most immunological active or the most immunogenic cancer is renal carcinoma with a very high tumor coating load in patients. And we looked at other cancer, fibrosarcoma was very low, and ovarian carcinoma was somewhere in the middle. So some patients have high level of a coating, some have low level of coating. So we took this data, and first we repeated what Brad Nelson showed. We looked at plasma cells and B cells in the tumor and found that it correlated with patient survival. And then we had our own new analysis of looking at tumor coating, and we found that if the tumor is coated, the chances of the patients to live longer is also bigger. So Brad showed the B cells and the plasma cells. We showed antibody coating, which is correlated with patient survival. So that looked interesting for us, and wanted to learn more. So we look for B cells in the tumors, and we use simple imaging approaches. And in some tumors, we can find germinal centers. In some, we did not. But in all of the patients, we could find plasma cells. You see here in green. We found plasma cells that surround the tumor, sometimes inside the tumor mass, but it's mostly in the connective tissue around the lymph node that we could find plasma cells. So we wanted to know if these plasma cells make anti-tumor antibodies. So we sequenced the plasma cells. I'm not going to show you, but we found clonal expansion and interesting data about these plasma cells. And we took the sequence of the antibodies and we produced it in the lab. And we produced monoclonal antibodies in the lab. And then we took primary tumor cultures, which we also made in the lab. And we have tumors, we have antibodies, we put them together. And some antibodies don't bind the tumors, but we found many antibodies that do interact with the tumor, that show positive binding. And that taught us that some of the plasma cells within the tumor can make tumor-reactive antibodies. Some of the plasma cells are specific for the tumor. And that was exciting. But then there was another finding. We made another finding that was really even more exciting for us. So in my lab, everything goes back to the germinal centers. So we had mutated antibodies, and we could revert the germinal center response by making the mutated antibodies in their germline form. So we take the mutations out, and we keep the V gene in the form of the germline configuration. So we make monoclonal antibodies, mutated and unmutated, and we test tumor binding. And the mutated version of the antibodies bind the tumor, but the non-mutated, in most of the cases, did not bind the tumor. So here we could prove that the germinal center contributes to anti-tumor or to tumor binding of these antibodies. The germinal center plays a role in anti-tumor immunity. And that was very exciting for us. And that's a proof that the Germinal Center is important in making specific antibodies against the tumor and allows tumor binding. And there are a lot of other results in the study. We also found a target, but the bottom line, what we found is that Germinal Center contribute to anti-tumor immunity. So we did this study, and I spoke about plasma cells from the tumor, but and then again, I gave a lot of talks. Like I spoke a lot about this project in many meetings, and I always get this question like. We spoke about intratumoral B cells, intratumoral plasma cells, but what about the lymph nodes? Like Germinal centers usually takes place in lymph nodes. B cell immune response takes place in lymph nodes. So what's going on within the lymph nodes? And we could answer this question because we collected also the lymph node from these patients. And so the question that we ask, what is the contribution of tumor draining lymph node to antibody-mediated immune response in ovarian cancer? And this was the PhD question of Nachi Nathan, a PhD student in the lab. So we started to study lymphoid organs of humans, and we need controls. So in human, you usually use one that wants to study human immune response in human germinal centers. Usually, scientists take tonsils. These are tonsils are removed from children, and you see many germinal centers stained by Ki-67. So this is a kind of okay control, but it's not age matched, and the tonsil is not a draining lymph node, so it's okay control, but not ideal. So we look for additional controls for our experiments, and we decided to take mesenteric lymph nodes from patients with IBD, and this is a true draining lymph node, and also now the patients are more age matched to the patients that we studied, the cancer patient, so that was a better control for us. So you can see the germinal centers over here. I already showed this picture in the previous slides. So this is the controls. So now we took the same staining and we analyze patient lymph node. So we take draining lymph node from the patients, and this is how it looks like. So what are you seeing here? You're seeing B cells, CD20. You see many tiny follicles within this tissue within this lymph node. You don't see proliferating cells. There are nearly no Ki-67 cells. You don't see here germinal centers. There are some macrophage here, which I'll speak about later on. But basically, we couldn't find germinal centers in the patient's lymph node, which was a huge disappointment. Okay, so there are no germinal centers. Nonetheless, we decided, okay, let's try to understand what's going on here. What kind of cells are these B cells? What are they doing? So we took these lymph nodes and we made a single cell RNA-seq. And we did also, when we did single cell RNA-seq, we couldn't find activated B cells or very few activated B cells. We couldn't find germinal centers. Mainly what we found is some naive cells. You can see it. You can see them over here. They are IgD positive. So these are truly naive B cells. And most of the B cells were memory cells, different kind of memory cells. They don't express AID, so these are not germinal center cells. They rarely express CD10, so also that they don't look like germinal center cells. They don't express plasma cells markers, and they express this memory marker. So these are truly memory B cells that reside within these lymph nodes. We found small amount of plasma cell. Again, a small amount of plasma cells. They are positive for XPP1 and CD138, but really these cells were very few. It was a very small cell population. So the major cell population within these lymph nodes are memory B cells. And now I wanted to know if these cells are relevant to the tumor in some way. So we took the sequence of the antibodies of these cells and we expressed them within the lab. So we culture cells, we produce antibodies, and then again we took tumor cells, we took antibodies, we put them together, and wanted to see if these memory cells are tumor reactive. And this is what we found. We found that some of the tumor cells express antibodies that can bind tumor cells. And here you see example. So here you see a primary tumor cultures. This is an isotype control. It's an antibody against a virus. It's irrelevant. Here you see two non-binder cells, tumor. and antibodies that we produce from memory cells that recovered from the lymph nodes. And here you see binding cells. These cells express antibodies with potential to bind the tumor. So in all of the patients, we could recover some memory cells that show tumor activity, that they can bind the tumor. So here, like I think we for the first time show a new concept. We show cells that express memory markers, and also they have tumor reactive antibody, which suggests that these cells are some kind of memory cells that circulate in the patient body, and they have potential to respond to the tumor. So these are tumor reactive memory cells. This is something new that did not exist previously, that came up in this work. So we have tumor reactive memory cells, and now we wanted to know what's the connection to the tumor. So we analyzed the lymph node, and now what we did, we collected rare samples from patients in which we could get both the tumor and the lymph nodes, and we took B cells and sequenced them, and then we pulled the data together and analyzed the tumor and the lymph node cells together in one analysis. So when we examine tumors, we find lots of plasma cells, and I also showed you histological analysis that the tumor contain plasma cells, and these cells that we analyzed in the previous study. In some tumors, we can also find germinal centers, which we didn't find in the lymph nodes. So that's a kind of surprising and weird, because you would expect to have germinal center in the lymph nodes. In the ovarian cancer patients, we don't find them in the lymph node. We find them within the tumor. And again, we found lots of memory B cells. So there are different subsets of memory B cells that came up in these patients. So here we compare the tumor draining lymph node to the tumor. So in the tumor, we find atypical memory B cells. We don't find them in the lymph nodes. And also we find class switch memory cells. This is the second most abundant B cell population within the tumor, and it's the most abundant B cell population within the lymph node. So we have these cells, we have lots of these cells, and we thought perhaps these cells are connected in some way. So how can we prove that there is connection between these memory cells? So we have the antibody sequences. So we analyze antibody sequences from the tumor and tumor draining lymph nodes, and in all of the patients, we could find sequences that were recovered both from the tumor and from the tumor draining lymph nodes. So these are shared sequences that came up from both of these organs, and it's per patient. So if we compare a tumor draining lymph node from one patient and take the tumor from another patient, we don't get any clonal sharing or very few cells. So that's internal control, and that means that our analysis is real, and we truly find unique cells that are found both in the tumor and tumor draining lymph nodes. And then we can make evolution trees from these sequences. Here you see a clonal family of cells. You can see that most of the cells in this family are found in the lymph node, but nonetheless, we could recover these sequences, the same sequence with some mutations within the tumor. So you see that it's a shared family in which the clonal members are found both in the tumor and in the tumor draining lymph node. And this is a proof that they originated from one ancestor. There was one cell which participated in a germinal center, perhaps in the lymph node, perhaps in the tumor, and then migrated to the other organ. This is one example. This is another example where we saw most of the clone is located in the tumor, and a few clones were detected in the lymph nodes. And it's important to me to say that there is no contamination, because we have the organs separately, we sequence them separately, everything is done separately. So there is no way that we just contaminate with sequences from a different organ. It's all done completely separately. So this is a true result, and we found the same sequences in these different organs. And then we ask if these sequences are tumor reactive. So this is an example of one antibody. We found that some of these sequences show tumor reactivity. So that these cells have the potential to respond to tumor antigens. So here we prove there is a connection between the tumor and the lymph node. We don't know if they are moving from the blood. Most likely they do, that these are memory cells that circulate from the blood and can come from the lymph node to the tumor or the other way around. We can't ask this question in human, but nonetheless, there is a connection between the tumor and the lymph node, and cells are moving between these two sites. So now I want to go back to a problem that we noticed in the beginning of the study. So we looked at the lymph nodes, and there were no germinal centers. So I wanted to try to understand why there are no germinal centers in these lymph nodes. There are tons of antigens and very active immune response going on. Why there are no germinal centers? So for that, we collaborated with Liat Keren from the Weizmann Institute. She specialized in multiplex imaging, and she helped us do a MIBI-TOF analysis using multiple antibodies of the tumor-draining lymph nodes. And this is what we found. Like we see T cells in this lymph node, we see B cells in this lymph node, and then we found these DC sign positive cells within this lymph node. And you can see that there are many, many cells which are DC sign positive in the tumor draining lymph nodes. So we wanted to better understand what are these cells. We looked at other markers. Some of them are CD14 positive, and they also express CD206, which suggests that these cells are suppressive macrophage that invade these lymph nodes. So this was with MIBI, and then we stand for this sign tumor draining lymph node, and you can see these small B cell follicles, and you see that the lymph node is like full of macrophages that express DC sign. So we did this analysis, and this is suggested to us, so this has reminded us a different study that we had in 2022. So I'm going to pause a little bit, and I want to go back to this study. So in 2022, a PhD in the lab at DB Ram, she found that when she put bacteria into mice, the bacteria induce formation of monocytes in the bone marrow, and these monocytes migrate into lymph node and basically kill the germinal centers. And the result that we found in the cancer really reminded me this study that when monocytes go into the lymph nodes, it's not a good thing for germinal centers. Usually these monocytes come, and it's a program. It's a program that under emergency, or that when we get a sepsis or bacterial invasion, we don't think about long-lasting immunity. We don't need germinal centers. The monocytes just come and kill the bacteria, and we can wait with long-lasting immunity and protection. We just have to get rid of the bacteria in order to survive. So this is a mechanism that we described before, and we think that what we see in the cancer is a similar mechanism. Now there is no bacteria, but this mechanism was triggered and affected the B cell immune response within the lymph node. Now, we can't do the experiment that we did in mice, blocking formation of macrophage, blocking entry of macrophage. We couldn't do that in human. We tried to do something simple in vitro. We take macrophages or monocytes, human monocytes. We polarize them to different kinds of macrophages. We take B cells, naive B cells, and germinal centers, and we just put them together in a dish. And if we don't put them together, if we just take germinal centers and naive B cells, we can see the germinal centers, CD27, CD38 positive, we can see them in the dish. If we add macrophages, the germinal cells are just gone. You can see it over here. So it's all well controlled. It's a 2 days experiment. The germinal centers don't just die spontaneously. We detect them, and then if we add the macrophages, then they are gone. And this is contact dependent. So if we do this experiment in a trans well, you can see B cells without macrophages. When we add the macrophages in the lower part of the trans well, you see that the B cells stay. So it's a contact dependent event. And so these are FACS plots. We also could quantify that, and we saw reduction in B cells. In fact, all of the reduction in amount of B cells in the culture was due to reduction of germinal center B cells. So if we quantify only germinal center cells by CD38 and CD27, they are all gone when we add the macrophages. We also look at CD10 plus CD10 plus B cells, these are also germinal center B cells, then they are all gone, but the naive cells stay. So that suggests that macrophages has potential to remove germinal center cells also in culture, also using human cells. Okay, so now I'll summarize this part. So what I show you is that in ovarian cancer, the tumor-draining lymph nodes do not provide fresh B cells to the tumor microenvironment. The lymph nodes do not form germinal centers, and there are no fresh cells or plasma cells that move into the tumor. Nonetheless, memory cells can move into the tumor, and they have potential to respond to antigen within the tumor. And I show you that the presence of macrophages in the tumor-draining lymph node has potential to disrupt germinal centers, also in human settings. But the most important message here is that we found tumor-reactive memory B cells. They are circulating in ovarian cancer patients, and we think it's an important thing, but because if we can reactivate these memory cells, recruit them to the lymph node, and perhaps make them seed tertiary lymphoid structure, perhaps we can enhance effect of immunotherapy, and this might be beneficial for the patient to turn on tumor-reactive memory cells within the tumor cells of the ovarian cancer patients. Okay, so this is the summary of this study. So I'll mention the people that made the work. So I presented the work by Nachri Nathan, a PhD student in the lab. He studied lymph nodes in cancer. I also spoke about Roy Mazor work, a study that we already published about B-cell immune response in the tumor, and that was a collaboration with Verit Sagi. I also showed Jing Liu study and also Liat Stoller-Barak study. They studied B-cell immune response in the nose. I should also mention our clinical collaborator Ram Eitan and Oded Raban. They give us tissues from the clinic. And these are the funding agency that support us. And that's it. This is what I had to tell you for today. Thank you.
Okay, thank you, Ziv, for this wonderful, illuminating talk. And I should just end the session by reminding all of you that you must ask Ziv all your questions on the YouTube channel. So please use that QR code that is in front of your eyes right now. Thank you, Ziv, and thank
Thank you.
you.
It was a pleasure. Thank you.